Hello friends, welcome you in Research Needs. In this video today, study of antihistaminic drug with the help of histamine chamber, mast cell stabilization method. So now we will enter into the Expharm software. We will get this type of interface and we'll click to the visual practice histamine chamber. To start the experiment, we will click here. So now after entering, we will see here equipment which name is histamine chamber. So what is histamine chamber? What it consists of? A histamine chamber is a specially designed apparatus used in pharmacology experiment to study the effect of histamine and antihistaminic drug, especially in guinea pig. It mainly consists of, here you can see, airtight transparent chamber, nebulizer and aerosol generator, air compressor pump, drug administration port, observation window. It is already the transparent acrylic glass we can see here. Outlet port. From above side we can the accumulated histamine aerosols that we can release by removing the covering from the above side. So this was the about the equipment. Now we will see the introduction. So now we will introduce what is mast cell. Mast cells are immune cells derived from hematopoietic stem cells in bone marrow and are mainly found in connective tissues, skin, lungs and the GI tract. They contain granules rich in histamine, heparin and various cytokines which are released upon activation. Its primary function is to mediate immune defense against pathogens and to regulate tissue homeostasis. Mast cells play a key role in allergic reactions by releasing histamine during IgE-mediated hypersensitivity, leading to symptoms like itching, swelling and bronchoconstrictions. They also contribute to inflammation by releasing pro-inflammatory mediators that recruit, activate other immune cells at the site of injury or infections. So after this we will see here in the title we have seen mast cell stabilization method. So what is mast cell stabilization? So we will see that. So the mast cell stabilization method is an experimental technique used to evaluate drug that prevents or reduces the release of chemical mediators like histamine from mast cells. Mast cells, when activated by allergens or other stimuli, degranulate and release histamine, leading to allergic reaction such as bronchospasm, rhinitis and inflammation. In this method, animals, generally guinea pig, because they are very sensitive to histamine, are exposed to allergen or histamine-inducing agents. Mast cell stabilizing or antihistaminic drug tested to see whether they can inhibit mast cell degranulation and prevent histamine release. Protection is assessed by observing outcomes such as delayed onset of bronchospasm, reduced allergic symptoms or intact mast cell under microscope. In short, this method demonstrates how drugs such as cromolyn sodium and ketotifen antihistaminic stabilize mast cells, thereby preventing histamine-mediated allergic response. Now, after this, we will see what happened in this experiment. The title is Study of Antihistaminic Drugs with the help of histamine chamber, mast cell stabilization method is performed using guinea pigs to evaluate the protective effect of antihistaminic drug. In this method, animals are exposed to histamine aerosol inside the histamine chamber, which induces bronchospasm, respiratory distress. Antihistaminic drugs are tested for their ability to prevent or delay the symptoms of stabilizing mast cells, inhibiting histamine release. This model helps in understand the pharmacological action of antihistaminic agents and their therapeutic roles in allergic conditions like asthma or rhinitis. So now we will see the experiment, how it performed. To start experiment, click here. So we will click here. Next, click here. Now you can see here the guinea pig inside the chamber. So we have to keep both sides. To proceed further, click here. So we will see the instruction first. Animals are divided into two groups. Administered one with an antihistaminic drug and the other with normal saline by the suitable routes. Put animals in histamine chamber and provide animals the exposure of histamine. Record the observation. So here we have two guinea pig. One is vehicle treated and one is drug treated. So now we will administer the drug, antihistaminic and normal saline. So we can give IP dose to these animals. So now we will click the animals. So while we will click, they both enter both sides. Once one animal this side and one animal this side. So now we will click, because the one animal drug treated is given antihistaminic drug and this it is given with saline only. So now we will click animals. So both are in both side chamber. So one is vehicle treated, so it is vehicle treated and drug treated. Here you can see it is showing some reactions just because of the histamine. Whole chamber have aerosols of histamines and it is not showing any reaction because antihistaminic drug is given to this animal. So it is showing the shaking reactions and it has a bronchospasm also. I think it has some problems. So now we have to write here allergic response. So we will write in vehicle treated yes, in drug treated no. So it has no any reactions. It looks calm. So now we will go ahead, see the observation. The vehicle treated allergic response yes and drug treated no. So now we will see the results. On exposure to histamine saline treated animals, guinea pig shows allergic response, and whereas antihistaminic anti-allergic drug treated animals, guinea pig, do not show allergic response. So this was the experiment. This experiment is very small. So how was the video? Please do comment. Thank you for watching Research News. Thank you very much.